which is hosted by the Yale Center on Climate Change and Health. So today we have a hybrid seminar, due to the COVID pandemic. We have the students joining us in person, but also for the students who cannot join us today, and also for online audience. Before we move on, I just want to have two quick rules, kind of housekeeping rules. For you guys have submitted the questions to our speaker. So at the end, we'll have a Q&A session. So you guys feel free to ask your question, raise your hand so the speaker can actually hear you very clearly. And for the folks online, if you have any questions, so please don't hesitate to put them in the chat box. And we will also go to them ask on behalf of the attendees. So it's my great pleasure today to introduce our first speaker of the seminar series, Dr. Benjamin Zaitchik. Dr. Zaitchik is a professor in the Department of Earth and Planetary Sciences at Johns Hopkins University. His research addresses hydroclimatic variability, including fundamental work on atmospheric science and hydrological processes, and application to problem of water resources, agriculture, and human health. So Dr. Zaitchik is actually also the president of the GeoHealth Section of the American Geophysical Union, in short, AGU. So another thing I want to mention is Ben actually got his PhD from Yale in 2006 from the Department of Geology and Geophysics. So we are very pleased to welcome back Ben at Yale, although virtually. So without further ado, let's welcome Dr. Benjamin Zaitchik.
Great, thanks so much, Kai, and thank you for the opportunity to speak. I have to admit, I've somewhat enjoyed this remote world and our ability to talk and interact at a distance, but I was a little disappointed when I'm not able to be up there in New Haven right now, because it would have been fun to come back. As Kai mentioned, I did do my PhD there, but not in public health, kind of cross 34 on Science Hill in geology and geophysics. But while I was there, I was not yet working in the GeoHealth area, but I got to see a lot of collaboration going on, particularly between Derlin Fish and some of his students in public health and my geology department, which really was my first exposure to this idea that you could really make use of some of our environmental information analyses to inform infectious disease analysis. So the talk today, I'm going to be focusing on malaria in the Western Amazon. I've got a long list of names here. That's only a partial list. I want to particularly acknowledge Bill Pan at Duke University, who has led most of the work I'm going to present on today from the epidemiological side, as well as Mark Janko, Cristina Ricalde, and Francesco Pisatuti, whose results I will be showing. So I might start with some deep background and perhaps an apology in that the idea that malaria is somehow an environmentally mediated disease is not particularly new, right? It shouldn't come as a surprise to anybody. In ancient times, malaria was associated with the rise of Sirius, the dog star, which would come in the days of mid to late summer around the Mediterranean, where the Greeks and others were studying this and aware of its impact. That is why we call them the dog days of summer, because it's when Sirius became visible. And you can see writings about this across the Mediterranean at the time. Hippocrates, who was famously very interested in the relationship between environment and meteorology and health, wrote specifically about how these cyclical fevers that we now understand to be malaria were associated with the season, and clearly understood quite clearly that this was not an astrological phenomenon, but that this was a phenomenon tied to the seasonality and to the oppressive heat felt at that time. Now, this is millennia before the mosquito-mediated pathway of malaria transmission was confirmed, and as well as before the Plasmodium was identified as certainly as the parasite. And yet this understanding that malaria was sensitive to these changes was clear. And the very fact we call it malaria, right? Bad air. It's the disease that is most associated inherently in our naming system with this idea of environmental sensitivity. And so you might think that we had this kind of figured out, right? So why in the year 2021 am I here to talk to you about our, attempts and our struggles to continue to understand in a predictive fashion the way in which malaria responds to environmental variability? And I think the answer is that it's a bit complicated. And so what I'm going to talk about here is something where we really need to understand the environmental influence, the climatic influence, as well as other environmental influences in the full context of a coupled natural human system that evolves with time. And so simply understanding that malaria has the potential to be sensitive to environmental factors is not in and of itself a useful or actionable predictive system. So talk today, I'm going to start off with some background on malaria in the Western Amazon, and apologies in advance if doing so is insulting to folks in public health who have a deep understanding of malaria in this region, but I'm not sure of everyone's background. So I'll go through a little bit of that history and current dynamics. Then I'm going to spend a little bit more time than you probably want me to on physical geography and hydrometeorology, because that's really what I bring to these set of analyses. Then I'll move on and just give 3 of the cases in which we try to integrate these kinds of environmental information systems to our understanding and forecast of malaria in this region. And I want to emphasize something that Kai said, was certainly type into the chat if you would like to say anything. Also feel free just to unmute and interrupt if I say something that is unclear. So again, basic background of malaria in the Amazon. This is from the Malaria Atlas. And what we see here is that the dominant type of malaria will be vivax, that is present throughout the Amazon basin, but we'll also see falciparum in some concentration. And in the Western Amazon, that part of Peru and Western Brazil that I'm going to be focusing on, you will see both in significant amounts. I should note that I'm zoomed in here on the Amazon basin. The Amazon is home to over 90% of malaria in the Western Hemisphere. And so it's really, in terms of studying the Americas, it's the place that one would want to be focusing a lot of effort on malaria reduction. And in this region, malaria is classically associated with deforestation, encroachment into the natural forest. So it's just a satellite time lapse showing over about 30 years, what we all know to be true, this massive deforestation. This particular lapse rate is from Brazil. You'll see similar things throughout the Amazon basin. Classic pattern here is a road gets built. You might have seen that flash across the screen at the beginning of the time series. Once the road is built, you get this herringbone pattern of deforestation as land is cleared for logging, but then also for agriculture and ranching. And this dynamic was associated with a massive burst of malaria in the Amazon region, particularly in the 80s and 1990s. And so that was really the time of the most rapid deforestation going on over much of the Amazon. Continues to be a major issue today, but that's when the rate was the highest. And what you had there was a situation where epidemiologically naive populations were entering into a region with where the Anopheline mosquitoes, the dominant vector of malaria, or the vector of malaria, were present in large numbers, and the kinds of livelihoods we were seeing in particular, this kind of entering the wilderness for logging and such, and then a lot of mobility going on, all led to this really strong epidemic peak. And from observing this dynamics, what now we would consider to be a classic hypothesis emerged called the malaria frontier. And so you have frontier malaria in situations where you have populations that do not have immunity and who do not have behavioral patterns associated with trying to avoid malaria because they're new to the area, enter into the wilderness frontier. And you get this burst of epidemic peaks, followed by a gradual adjustment as you get some resistance building up, as you get populations behavior changing, and as you get livelihood changes that maybe are a little less mobile and include less interface with wildlands, and you settle into an endemic pattern, this endemic malaria. And so, this is flashed through enough times that maybe you've noticed by now that you can kind of see the timing, right? While things change throughout the time series I'm showing, after about the year 2000 or so, the change isn't as rapid. You aren't seeing as much clear cutting, right? That mostly happened in the 80s and 90s. Again, this is a time series from Brazil. You'd see similar things in the parts of Peru and Ecuador that we're focusing on. So when I talk about malaria today, I'm going to be focusing on the last 20 years, which is really post-frontier malaria. Okay, so this is the time where we say, okay, we've kind of been through that initial burst of malaria that happens when you enter the frontier. And now we're in the situation where we are looking at transmission patterns in populations that I wouldn't say that it's a stable population. There's always movement going on, but you're no longer talking about this encroachment. You're talking about interfaces within what is more or less a settled area. Okay, and so what does that look like if you just look at case numbers in the Amazon? So here I'm showing a time series from 2000 on, and so what you're missing over to the left here are the really high numbers that preceded this. So the numbers on this curve, you can kind of see Brazil, that red curve coming down, right, from what was a really big peak in the 1990s. And if you ignore Venezuela, which as we all know has had its own challenges, you would generally say, oh, this is kind of a story of cases falling, okay, from that frontier malaria peak. But if you look a little more closely over the last 20 years, you'll see that progress has stalled and even reversed. And so expanding the y-axis a little bit here to look at Colombia, Ecuador, and Peru, just over the past 15 years or so, what you see is a rather significant peak in Ecuador. It came down a little bit after that, but it's come back up. Peru, quite a significant percent-wise increase because the cases got so low in the mid-2000s or the early 2010s. And another significant, sorry, that was Ecuador. Big significant increase in Ecuador. I misread my labels here. Then the bottom one is Peru, showing the significant increase again. And so you see these large percent-wise increase in these Western Amazonian countries. Focusing on Peru specifically for a moment, because that's where a bunch of our data are going to come from that I'm going to show in the next section. What you see here is a phenomenon where, again, cases were quite high in the 1990s, but there seemed to be a period where you were at a kind of a stable level in the 2000s, and then a rapid decline to the point where it was really getting close to elimination around 2010 before it burst back up. And so now what's been happening? So that period, as I'll get to it towards the end of the talk, was a period of a significant intervention, an attempt to eliminate malaria from this region. So the PANAFRO program, which ran for about 5 years, involved a number of malaria control activities. Again, details come later, and it really did seem to work. Then in 2011, you had this historical flood, okay, the flood of record over much of the Amazon, I mean, the biggest one in the observed record. And it had tremendous impacts across the region. But one thing that happened was, well, we saw a burst in, or an increase in malaria cases, this reversal. Okay. Now this flood coincided with the end of the PANAFRO program. And so we have some disentangling to do about what's going on when it increased. And when this first happened, there was a sense like, okay, a flood happened, there's going to be a bunch of malaria and then it'll come back down, but it didn't. It just kept going up and up and up. In the time since that flood, you've had several other destabilizing events. 2015, as you might be aware, was this mega El Nino with global effects. You also had dengue and Zika, in particular the Zika scare coming through this region at that time, which really stressed health systems. And so one thing that we're trying to do now is say, okay, in this context of intermingled climatic effects, social effects, epidemiological effects, what exactly is going on here? And this is critical because, 10 years ago, if I were giving this talk 10 years ago, we'd be talking about elimination of malaria in the Amazon. We are not talking about that right now. We're talking about trying to control what seems to be an increase, and though you don't see it on this graph, because Peru seems to settle down a bit, not just an increase, but really maybe a significant continuing increase of malaria in the region. Okay, so let me jump into the physical geography and hydrometeorology of the problem. So let me start off with a little bit about the vectors. So as I will attempt to stress throughout this talk, when we talk about the influence of environment and hydrometeorology, we're not just talking about mosquitoes, okay? Mosquitoes are a big part of it. And so that's why I'll start off with them. But we always want to be thinking about mosquitoes, we want to talk about the pathogen, and we also want to talk about human behavior. Nevertheless, the influence of land cover and hydrometeorology in particular on Anopheline mosquitoes is going to be a big part of our story. So I want to make sure you're familiar with what's going on in the Amazon. So the red here is showing Anopheles darlingi. That is the dominant malaria competent vector in the Amazon. There are a whole bunch of others, a great diversity of Anopheline mosquitoes here, but the darlingi is going to be the #1. And if we zoom in a little bit, so I just have a little box there around this portion of the Western Amazon centered on the Loreto district of Peru, which is kind of the northern Amazonian district in Peru. You can go and study this there because a lot of really good work has been done by some of the members of the team that were on my title slide and people who preceded them or partnered with them in this area, doing really strong work on mosquito surveys. We're collecting, we're doing species typing, and this happened along various areas in the region. And I don't know how well this is showing up on your screen, but that red inset there is a Landsat satellite snapshot of the area, and you might see red dots, yellow dots, green dots. Those are all showing collection sites where breeding habitats and mosquito species types were collected at larval and adult stages. And they were organized along transportation corridors, these surveys. And so the red dots are along a highway that connects Iquitos to Nauta, a town to the south. The yellow dots connect Iquitos to Mazan up in the north. And then the green dots are going along various rivers that are used as transportation corridors. Let me just zoom in on that a little bit so you get a sense of this region. So here, this is just a kind of a true color satellite image of what I showed in that previous slide. So you see the Amazon River flowing south to north here through the region. That urbanized area that you see in the along the banks of this meander is Iquitos. Iquitos is famously the largest city in the world that you cannot reach by road. You either have to come in on the river or you have to fly in. The rivers are the dominant transportation networks, but we have these roads I showed before. There's one to the north that kind of cuts off here going to Mazan, but this highway here, the Iquitos to Nauta highway, is kind of the biggest road in the area. And you see that herringbone deforestation coming along that road. Okay. And so what we have here are mosquito collections in an area of land use contrasts, including the pristine forest, as well as grading into areas of significant agricultural activity and urban activity. And so we can then use our satellite images to classify the different types of cover we see here. And these range from different water types. We always want to distinguish between clear water and silty water in the Amazon. They're very different ecologies. And then different kinds of Amazon basin land cover type, including the anthropic types, such as disturbed vegetation and bare ground and roads and buildings, and the natural vegetation types, including different types of forest. Okay. And so when we analyze these together, the land cover information with the mosquito information, you find some interesting patterns. And what I have here are all Anopheline species. Okay, I didn't bother spelling out all of the species names because they're long and it doesn't matter too much. But what this box plot is intended to demonstrate is that as your forest area decreases, okay, as you go to the down on the y-axis into the negative area here, you will see different relationships with different species. Okay, and so when you have a, sorry, I apologize, let me step back. The y-axis here is the association, okay? And so you see negative associations between forest area and some species and positive associations between forest area and other species, okay? And so what's interesting about this is that you say, okay, so it's gonna be changing species assemblages as land cover shifts from natural forest to more cleared area. But it's somewhat systematic in that the species here over to the left are the malaria competent species. And you'll see Anopheles darlingi here on the far left. And so that's a dominant vector, and all of these others are vectors also. These are not, okay? So it so happens that as you clear forest, you might not actually see an increase in the total number of Anopheles mosquitoes. You often will see a decrease in the total number of mosquitoes of all species, but you'll see an increase in the prevalence and the absolute number of darlingi, of your vector species. And in fact, this can be quantified using the data we had. We found that for every 1% increase in cleared land area, you have close to a 4% increase in the odds of finding an Opales darlingii at a collection site. So we have here is human-wildlife interface causing more mosquito-human interactions, and also the anthropic disturbances of the landscape increasing the proportion of your competent vectors. Okay, so this is a recipe for increased malaria transmission. So this is a fairly detailed study that we could only do in places where we had really detailed entomological collections. We don't have that everywhere, but at least from the satellite perspective, we can take this kind of lesson done at high resolution and zoom out a bit. And so as we try to look across all of Laredo states, so this is showing Laredo state of Peru, and this analysis has now been extended to include the Amazonian portions of Ecuador, as well as parts of Colombia and Brazil. We can make use of satellite data, and here I'm showing the MODIS satellite data. If you're not familiar with MODIS, it's a NASA-supported mission that's been up for about 20 years now. And unlike the previous image that I showed, which was a Landsat, higher resolution, 30-meter resolution, but you only get snapshots every once in a while, MODIS is giving you 250 to 500-meter resolution, but it's giving you daily images. And these really cloudy areas, that's important, right? Because you need to catch when you can a view through the clouds. And that allows us to use phenology, that is the seasonality of the vegetation, to do a more detailed classification of land cover types. And so on the left, you'll see a classification we did using MODIS. We can then, because the satellite's been up for 20 years, look at change in these forest types over time. All of that can go into our malaria risk analyses. And on the right, what I'm showing you is a product that I did not develop, but that NatureServe developed, which used a combination of satellite data and measurements on the ground to come up with ecological systems that we view as potentially relevant to malaria. In particular, the red areas on this map are areas that are forests that are flooded by what they call black waters, so those tannic waters of the Amazon. And then in the light green, you'll see other areas that are flooded by what they're calling white or clear water. It might have sediment in it, but it's not tannic. Okay, so again, different water quality, different ecology. Okay. So what I've taken here is land use, looked at really high resolution land use to understand mosquito distribution, used different satellite assets in order to zoom out and say, well, what can we say at scale about land use and vegetation types? And then also, thanks to the NatureServe analysis, link that somehow to hydrology, right? Because now we're talking about ecological zones that are defined in part by their flooding regime, which is a key consideration in the Amazon, right? There's a lot of forest that's different from other forests, and much of that has to do with these flooding regimes. Okay. So this brings hydrometeorology into the picture, right? And so how does hydrometeorology matter? As I mentioned, it's going to affect the vector, right? We're concerned about breeding sites, survivability at different life stages, the life cycle, speed of the life cycle of the mosquito, dispersion of mosquitoes influenced by winds and temperature. And so wind, temperature, and certainly precipitation and moisture conditions in the soil and surface puddles are going to be a big deal. We also know the plasmodium has temperature sensitivities, and that the vector's competence to transmit the plasmodium is a function of temperature. Okay. On top of that, you've got human behavior. And so migratory labor, in particular logging in this area, is very sensitive to the river height, because you need the rivers to be at a certain height in order to float the logs downstream. So that will have an influence. And then, of course, agricultural activities will be sensitive to the seasonality of hydrometeorology, as well as to interannual variability. When you get to interventions, you also have an issue that anyone who's worked in malaria knows, which is, will people use bed nets? And when it gets really hot, very often it gets harder to comfortably use a bed net. So how are we going to do hydrometeorology? So there are a lot of different ways you can do this. The system that my group uses, and kind of one of our major contributions to this malaria problem, is called the land data assimilation system. So that probably doesn't get discussed too much at schools of public health, which is appropriate. So let me give you a little background, because this is an area where any of you potentially working on various climate environment influence on disease, but really any host of public health issues might be able to make use of such a system collaboratively or on your own to really bring environmental data in a powerful way. So what an LDAS does is it merges observations with numerical models in order to get your best possible estimates of what's going on at the land surface and the lower atmosphere, kind of your surface meteorology. Why do you do this? You do this because satellite observations are amazingly powerful tools, but they're snapshots of single variables. And so if you want a comprehensive view of what's happening with all the potential variables of interest, you kind of want a model, right? You want something to give you spatially and temporally complete and consistent representation. But those models don't necessarily represent reality, particularly in data limited environments like the Amazon. And so what you do with an LDAS is you basically get the best of both worlds to the extent possible. You have an advanced physically based model that is trying to simulate what's going on with your weather and with your hydrology. And then you've got satellite observations that inform that model and kind of keep it realistic. And so in schematic form, what you have is a bunch of landscape information, such as the land cover analyses I've just shown you, often satellite derived. You have meteorological data, which is also often from satellite or from other weather analysis systems. Those all drive a numerical model, which is then going to produce estimates of energy balance and hydrology. Okay, so that'll get you at the the temperature, radiation, wind, moisture conditions you care about. As you run this model forward, you assimilate observations, and so you can update observations. So, for example, information about soil moisture variability, gridded estimates coming from satellite, can be brought into the numerical model to update the model's estimate of soil moisture. And so you end up with a system. This, as should be obvious because we're using update observations, this isn't like a future projection model, right? The model itself might be able to, but the LDAS system is retrospective up through real-time monitoring, where you're bringing in these update observations because the observations you can only have after you've taken the observation. Okay, and so these LDAS systems are in a lot of places. It's related, first of all, to weather forecasts, right? So weather forecasts use LDAS as well as assimilation of atmospheric variables. So those are used all the time. We also use these LDAS in a lot of the work we do, for example, in agricultural monitoring in the United States. Climate assessment reports are very often include LDAS, like the National Climate Assessment of the United States. Work we do with the famine early warning system in Africa. These LDAS are known to be pretty useful ways to get information. And so some of them have outputs that are available that you can just get because there's already someone running it. If you're interested in that, please contact me and I'll try to put you in touch. And then sometimes we run them ourselves to optimize them for a region we have here. So just couple more minutes on this, just so you understand the basic principles here. One of the most important starting points is satellite-derived rainfall. We're using a couple of products here. I'm not going to bother with the acronyms because they don't matter. They're there in case anyone attending today is from the satellite world and is interested in what we're using. Okay, so CHIRPS and GPM iMerge. We then use that MODIS satellite that I already described to get our land cover and vegetation characteristics. And this cartoon here is showing you our model. It's called the NOAA Multi-Parameterization Land Surface Model. And what it's doing is it's simulating multiple layers of the soil, different vegetation types, shallow groundwater. We also work into it a downscaling routine to get better surface meteorological estimates. It doesn't simulate the atmosphere, but it can help to downscale atmospheric conditions. And it also does snow, which matters. It actually does matter to us because we want to get the runoff coming out of the Andes, but it doesn't matter locally in the Amazon, obviously. So that's all kind of for the local energy and water balance solution. We use NOAA MP. We then couple it to a river routing model called HIMAP. And HIMAP, the Hydrological Modeling and Analysis Program, that's what that stands for, allows us to model things like the floodplain. And that's, as you can imagine, is really critical when you're talking about mosquito habitats. So we get the river heights, we also get the river width and the area of flooded river boundary at any given time. We run this at 5 kilometer gridded resolution, 5 kilometer by 5 kilometers, so 25 square kilometer. And then around Iquitos, that city that has a large population center, we nest into 1 kilometer for some higher resolution analysis. As we run the model forward, we can take advantage of these assimilation capabilities. And depending on, we run multiple simulations for different purposes. Sometimes we might be assimilating satellite-derived estimates of soil moisture or leaf area index or water storage, terrestrial water storage, just meaning all the water stored in the soil column and groundwater. These are all observables at different resolutions from space using different civilian space missions. And those will all help to improve the performance of our model. And then you can get an output like what I'm showing on the right-hand side of the screen here, which is just a standardized anomaly in soil moisture, showing a period where in our area of interest, for example, there was some drought going on in the northwestern Amazon, as shown by a negative standardized anomaly in soil moisture as captured by our system. Okay. This is actually, I'll come back to this in a moment, but this particular snapshot is an interesting example, and that's showing what might be considered the classic El Nino pattern. Okay, so it's an old snapshot. This one's from 1998. I accidentally cut the date off of it. This is the monthly anomaly from a month in 1998. And what you're seeing here is the 1997-98 El Nino bringing catastrophic flooding to the coast of Peru and Ecuador and drought to the Amazon basin. Okay, again, I'll return to that in a moment, but that's kind of a classic El Nino pattern in the region. And so here's just a quick animation to show what you're getting through time. I'm showing monthly outputs here. In fact, we get hourly outputs from the system that we can then extract for different geographies to perform our malaria analysis. Information on things like your air temperature anomaly, your rainfall, your soil moisture anomaly, your runoff, your river height, et cetera, et cetera. Okay, and so this is all the information that we're going to be bringing in, combining with the land cover and ecological information to try to get this environmentally informed malaria analysis and early warning systems set up. So one thing that you might be wondering is, okay, I just mentioned this was a data scarce area, right? And that these are outputs of some system that's combining satellite data with its uncertainties and a model with its own uncertainties. How good is it, right? And can you trust it? And the answer is that in any study you do where you want to make use of this kind of environmental data, you want to make sure that either you or someone else has done a good, clean analysis of how well that system performs in your region and season of interest. Okay, you don't want to just take this off the shelf and say, "Oh, I know this is going to be fine where I am." And so we've done some analysis. I'm not gonna make you sit through our whole analysis. It's the kind of thing that we spend our days, nights, and weekends doing, right? Make sure these systems work well and trying to fine tune them. But we have some data here that Christina Recalde, a PhD student working with me, has from Ecuador and some data from Peru, looking at things like, okay, how well do we do in observations in blue versus our model simulation on rainfall? And there are good and bad things if you stare long enough at this chart. It's like, yeah, we're getting about the magnitude of rainfall is not bad. The seasonality is pretty good in most places, but then you'll find there's some wet and dry biases in different places that we're investigating. Similarly, you can then look at the soil moisture. Soil moisture is harder because rainfall, there actually are rainfall observations. Not many, but there are some, right? Soil moisture, there's like basically no in situ observations in a consistent way in the study area. And so we have to use satellite data to compare it to. So here we're comparing this observation in gray, which is really a satellite observation, with our model simulation. And again, seeing some good, some bad. Here we really do have to question the fidelity of both the observation and the model, since the observation is satellite derived. But this gives us a sense. Do we have a consensus across our different estimates as to what's going on here? And this is tricky, right? Because getting soil moisture right in a complex hydrology like the Amazon is no trivial task. So this is an area where we spend a lot of our effort. Last point I want to make on this physical hydrology, hydrometeorology, before finally getting to some actual malaria results. It's really important whenever you're doing a study like this, right, to distinguish between when I say that there's hydrometeorological variability, am I talking about geographic variability, wet versus dry places? Am I talking about seasonal variability, right, a wet season versus a dry season, for example? Or am I talking about something like interannual variability, like, oh, we had a drought year, or we had a year with more flooding? And that's really important, first and foremost, to understand process, right? You want to know that if you get a statistical result that comes out of throwing some environmental variables into your model, they're significant. What is it that you're seeing, right? And also, is what you're seeing a proxy for something else, right? If you see if you classically see like, oh, there's a wet versus dry season response, or a warm versus cold season response, when I look at the cases of malaria, is that because temperature is affecting malaria, or is it because there's a seasonal cycle in temperature and seasonality for some other reason is affecting malaria, and I'm calling it temperature? Okay, and so you want to be clear on whether you're looking at the geography, the season, or the interannual variability. And this is on my mind a lot these days, both because I do a lot of this work, and as I know Kai appreciates, and probably others in the audience as well, there's a lot of conflation of these things in the COVID-19 literature with different claims or attempts to claim environmental sensitivities. Some really good work, right? But also a lot of these kind of naive, I would say, studies that came out showing correlations or associations that were simply showing a seasonality or, you know, a coincidence of two patterns, the whole correlation versus causation problem. That I think part of the problem there was a misunderstanding or a misframing of what kind of climatic variability we're talking about. Okay, get off that soapbox and simply say for that third thing, all I've shown you here is seasonality and spatial variability. I haven't shown you interannual variability. And I want to comment a little bit on that in this region because anyone who's worked on malaria in the Amazon or other malaria zones probably are aware of a lot of studies, good studies, right, that have associated malaria with various large-scale climate modes. These drivers of variability, okay? And so the big one is El Nino, the El Nino Southern Oscillation, okay? But there are many others. It's an alphabet soup that I won't get into. El Nino in this part of the region, so one might well expect an El Nino effect here, right? It's called El Nino because of the effects it had. You know, it was first characterized on the coast of Peru and what it does to the sardine fisheries off the coast of Peru. And so this is kind of like the home of El Nino, right? And so we should certainly expect an El Nino effect. And as I showed a few slides ago, a classic pattern would be high rainfall on the coast, drought in the Amazon for dynamical reasons that I won't get into. It's not that simple or that predictive as a simple univariate association in this part of the Amazon, at least. There are some other parts of the Amazon that respond a little bit more reliably, but I'll tell you, it's always a little complicated, okay? But here, just taking again from Christina's work here, looking at El Ninos and La Ninas the past 20 years, and if it's red, it means you've got drought or drier conditions. If it's blue, it means you have wet anomalies. And again, during El Nino, we should be seeing red in the Amazon. And here, you know, here we got our Laredo state. Sorry, this is just Ecuador and Peru I'm showing here. So we've got this kind of, here's your northern Amazon portion of our study region. And what you're seeing is that, yeah, during some El Ninos, you do see that drought pattern, okay? But you also see it in this La Nina. And then there are some El Ninos where you don't see it all. And in fact, that big monster El Nino that hit in 2015 and how this affects globally, it was wet in our part of the world when you might have thought it was supposed to be dry. And so there are some complications here, okay? I'd like to say that one could use ENSO, El Nino, La Nina oscillations effectively statistically in a forecast system here if you accounted for enough other variables. Okay, I'm showing you, highlighting the fact that it's not enough of a predictor of rainfall in its own right, okay? But combined with other factors, you can probably get some skill. But we've decided to take a different approach, which is rather than using these kind of teleconnections, these like remote connections to El Nino directly in our model, we run a dynamically based forecast. And so what we're doing there is, again, this one's a little detail for those who might be working at this interface of climate and health. We run what we call a subseason to seasonal forecast, you know, a few weeks out to, well, you can go to 9 months. We really are only going out to 3 months right now for this application. And what you do is you take what I already showed you in the LDAS, the satellite landscape analysis, run it through a land data assimilation system. That provides initial conditions from which you generate an ensemble. So your seasonal forecasts are these probabilistic ensembles, just like weather forecasts. And these are these global atmospheric models that we run forward. We run them forward using initial conditions of the hydrology locally and the ecology locally. We downscale the meteorology from those global forecast systems using some algorithms that again, I won't get into, but happy to follow up with anyone doing this kind of work. And then we put that into a hydrological forecast. We run it through the same modeling system. It's no longer data assimilation because we don't have observations. We run that system forward. Okay. So why do all of this? Because it pushes your forecast time horizon out. If I give you, right, or let me say, if I, as the climate guy on the team, give Bill and Mark, the epidemiology guys on the team, a monitoring system that is operationally saying what the moisture is right now, they can forecast malaria because there's a time lag, right? So they'll get a pretty good forecast because it takes time from what the signal that I'm sending them to propagate through the ecology and the human systems. But if I can give them a forecast of what's going to be like in 2 months, that gives them, 8 weeks more lead time, and you can make a different set of decisions given an extra 2 months, right? So it's all about this uncertainty time horizon trade-off here. The more we push out for a greater time horizon, the greater our uncertainty, but also potentially the greater power of the decision making that kind of system can empower. So how do these forecasts look? I'm not going to make you sit through a whole forecast skill analysis, but just want to make one point here. If you just focus, let's say, on correlation here for the sake of time, if there's hashing, it means statistically significant skill. And what you see here is that looking at something like soil moisture, we get really good forecasts for 1 month, and then it begins to degrade, particularly degrading in these wet areas. You maintain some forecast skill out in the dry areas because there's so much memory, right? If it's not raining much, most of the initial conditions that matter. But as you go out, we say the result here we might say is that we can really do a nice job of getting you an extra 4 weeks, right, on the system. If you want 8 weeks or 12 weeks, we're not going to be contributing that much skill from the forecast. And so it's important both to have the capability and to understand the limitations of the capability. All right, so we do all those analyses, and then this is not my work, this is the work that Bill led. He took all of this ecological and hydrometeorological analysis and did an objective regionalization, the principal components analysis on the variability, ended up with these 3 different factors that are loaded by different properties of the system, accounting for human systems as well as land use and hydrometeorological conditions. And from that derived these 7 socio-environmental regions. And the principle here is that these regions are reasonably homogeneous and regionally distinct from each other with respect to human and environmental factors. And also, as it happens, this was not necessarily integrated to that, but because you've included the human systems in the analysis, most of the travel stays within the region, and you typically have similar vector species within a region, okay, and similar livelihoods. So what we then say is that we're not going to develop one malaria risk model for, and again, this is now we're seeing Laredo region, so this part of Peru, we're going to develop a system that has customized models based on socio-environmental regions. So in the remaining time that I have, which isn't much, I know, so I'll touch on these lightly, but these are just examples of how we can pull this all together, all right? And so the first thing, kind of the motivation for this whole presentation, this whole project is forecast, right? And so using these socio-environmental regions, then aggregate malaria data, which we have about 300 health posts contributing data, passive surveillance. They get aggregated to a socio-environmental region, and then we try to predict whether there's an outbreak based on the Ministry of Health's definition of what an outbreak is, which is exceeding a certain threshold in terms of case number per population. Again, this work laid out of Duke by Bill, and he used this unobserved components model as a statistical method and was seeking to get a time horizon of 4 to 12 weeks. And again, because it's customized by region, what you'll find is that the model has different variable importance and is structured differently for the different models. So region 1, which includes Aikido, so it's kind of like our most urban area, we can describe that in terms of the characteristics of the socio-ecological region. And then we can say, okay, what explanatory variables from our environmental suite end up being significant? It turns out to be soil moisture. You can then look at a region like region 3, kind of really out in the forest, very low population density. It has a different description. It's going to have different statistical characteristics to this unobserved components model. And in this case, minimum temperature came out as the more significant variable. Both of these variables, of course, if you look at the literature, are used in malaria prediction. So they're both plausible, they're plausible pathways, but different ones came out as more predictive in these different regions. Okay, so then we run the system. We have to run the system starting 4 weeks before the present. Why? Because it takes about 4 weeks for surveillance to come in. Here's the percent of malaria of health posts reporting their malaria data. As you can see, this is the present. At the present, you have fewer than 20% reporting. If you go back 4 weeks, you have close to 100% reporting, which means that you have a good, previous cases are an important predictor of future cases. So the forecast includes a 4-week forecast of the past, and then we want to go out to 8 or 12 weeks in the future. We have this moving outbreak threshold because it varies seasonally and by location. What MINSA, the health ministry, decides is the right threshold to declare an outbreak. And then we might have an observation and a confidence interval around that observation. Just to give you an example of performance, 2016 was the first year we really tried this. So this isn't a systematic analysis, just showing you the kinds of things we look at: true positives, false negatives, false positive, true negative. For an outbreak in any of these ecoregions, looking at ecoregion 1 and 3 here, over the different forecast time horizons, our sensitivity and our specificity. In a nutshell, we do really well in ecoregion 1, fades a little in specificity as we get out to the 12-week time horizon, still pretty good. Ecoregion 3, we do not do that well. Okay. And so again, small sample, 1 year, but just our first test was showing us that we're gonna get different performance in different ecoregions. Okay. And so that's all at the ecoregion level. I'm not going to get into too many more results at that level right now, but rather say that to be decision relevant, we have to go down to the district level. So the lines here on this map are separating the districts, okay? And so the colors of the ecoregions, the lines are the district. We really want to be at district level. And so for this, again, we won't get into the details right now, but Mark Janko implemented this hierarchical Bayesian spatiotemporal logistic model, where you basically have your district outbreak probability being a function of the probability of an outbreak in the ecoregion that contains the district and some district-specific properties. When Mark downscaled and looked at some of these analyses and then did an evaluation over a retrospective period, these are the kinds of sensitivities and specificity we're getting for different districts within each ecoregion. Again, just showing ecoregion 1 and 3 here as examples. And you'll see that, again, pretty high variability. So we were doing well in ecoregion 1 at ecoregion level, but you'll see that, for example, in the district of Fernando Lores, there were some pretty significant errors in this retrospective period, and different kinds of errors in different places. So all stuff for us to look at. In ecoregion 3, kind of uniformly doing worse in general than ecoregion 1. So why is that? Why are we doing poorly in ecoregion 3? Multiple reasons. One thing I want to emphasize is that ecoregion 3 was very much located kind of up in this area. So first of all, malaria cases are generally low there in total because it's such a sparsely populated area, but it's also a border area. It's a border area that is transected by transboundary rivers. The transboundary rivers are the transportation in the region. And so what we find is that our model fits most poorly here in ecoregion 3, another ecoregion dominated by transboundary river, doesn't do well in places along the rivers, okay? And so that's one big weakness in the model that we're working on. And oops, these slides got reversed. And I just want to point out that that we are looking at, and we had a paper recently led by students. And so these are students from Duke, Johns Hopkins, Ecuador, and Peru, who took the initiative to really lead an analysis of this cross-border spillover. And that's something we're looking at now. Okay. So that's where the forecast system is. We brought it in 2019. We did some operational forecasts for the health ministry. It was all looking good. Then there's political change and COVID, so we're a little bit on hold right now, but we've got a system that we've proved we can use operationally. We continue to try to improve the performance. Policy evaluation. Okay, so I'm going to give one example of policy analysis we've done. That is a PEMAFRO, which is this project for malaria control in Andean border areas, active 2006 to 2010 or 11, depending how you count it. They did 4 kinds of things: long-lasting insecticidal nets, better rapid diagnostic tests and other monitoring tools, case management with antimalarial drugs and training, and environmental management for vector control. So they're doing these 4 kinds of things. And it kind of worked, right? So this is Vivax and falciparum and Laredo, and it sure looks like over the PEMAFRO period, the case counts are going down, 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 approaching eradication, which was the goal of the program. Then it stopped suddenly in 2011, cases start coming back up. And what we can do is we can leverage that district model that Mark Janko developed, right? Not only using it for forecasts, but then saying, well, let's include in that model structure the different interventions associated with PEMAFRO, because we know at district level and with monthly timing, what kind of interventions were done where. Let's integrate that to a model and then do an interrupted time series analysis and see what those interventions actually accomplished on the background of climate variability and all the other variables in our model. So kind of an environmentally controlled analysis of the effectiveness of the intervention. And what Mark's found is that, well, you can kind of quantify this. So the blue line here in the top left, top is Vivax, bottom is falciparum. Blue lines are the model, dots are the observation. On the left, we have the PEM-AFRO period, and we see that our model systematically, if you don't tell it about the intervention, systematically overestimates the cases in this period for both Vivax and falciparum. In the post-PEM-AFRO period, starting in 2011, quite the opposite. Our model has cases down here, the observed cases were much higher. Okay. And so take those together and come up with estimates that about 150,000 cases were averted by PEM-AFRO. That was the amount of malaria averted thanks to PEM-AFRO. And had you continued it for another 5 years, you would have averted another 150,000, not to mention the long-lasting impact of driving cases that low, right? And so here we have an analysis of both the effectiveness and the cost of removing a program without a good continuity plan. And then you can zoom in, because again, we have this district level information on each kind of intervention. I see I'm running out of time, so I won't spend too much time walking through these maps, but green shows incidence ratio less than 1. And so we can look district by district and say, okay, for falciparum and Vivax, for each of the 4 intervention types, environmental management, bed nets, et cetera, in which districts do we see the most effect when we add or remove this from our interrupted time series analysis? And there's some interesting patterns that appear that we're in conversation with some of our partners about to figure out what might be effective in the future. One other cool thing, just mentioned that you can do with this is try to figure out how much malaria and dengue there is right now in this area, because we have no idea. If you look at what happened in 2020 with surveillance, I mean, the health system basically shut down. And so it looks like it was a great year for malaria control, but of course it wasn't. So we can then use this same modeling approach to try to estimate how many cases there really were in the year 2020 and 2021. And as you can see, we estimate that there were at least 3 times as many cases. Okay, last point I want to make here is that I've showed you some malaria modeling cases that are process informed, but at their heart statistical, right? These are empirical analyses. And looking at intervention scenarios, we are also looking at explicit simulation of behavior, okay, to get these coupled natural human systems right. And the way that we are doing that, led by Francesco Pizzituti, is with agent-based modeling. And this is a kind of cool agent-based model Francesco built, in that it has agents that are mosquitoes, humans, and plasmodia. Okay, so you have all of these are agents interacting. And here is just an example of one of the villages where he's applied this, where you can have different households, and all these agents are interacting and influenced by the environment. In that here we see different kind of breeding habitats influenced by seasonal flooding, with information from our environmental analysis system, changing the hydrology. And then you've got the cases happening in these households, each of which is also experiencing its own environmental conditions. Okay. You can then run scenarios of control, for example, vector control strategies, one thing we like to look at. And so we're looking at here at one of these environmental control applications and saying, well, what if you do larval habitat control around a certain buffer radius around each household, right? How well do you do at 50 meters, 100 meters, 150 meters, 200 meters when you talk about malaria incidents, total Vivax falciparum? And the idea here is that by understanding in this agent-based model movement patterns, right, and the sensitivities of the different agent types, we can get a sense, say, well, really, you want to probably get out, well, you take your pick, but I would say at least 150 meters might be considered very effective. Anything beyond 200 is unnecessary. All right. And so this is parameterized for one set of villages. It's very data intensive, but nevertheless, I think it indicates a powerful way to use your environmental information in a different manner, not as an empirical predictor, but as a variable within a model in which different agents are responding according to decision rules to this variability. You can also use the same tool, and Francesco has, to look at the importance of mobility, right? So that's something people talk a lot about in the past couple of years, right? How much mobility influences disease transmission. It's an old story for malaria, But what you'll see here is that if you look at your observed black line here of the average monthly malaria incidence along the Napo River, first thing you know is that, well, okay, if I run this model with no asymptomatic cases considered in travel, you assume that no asymptomatic people are traveling, you way underestimate the incidence rate. So we know there's a lot of asymptomatic activity going on. And then we can say, okay, as the percent of traveling workers increase, we would expect the incidence rate to increase. And we're at about the right order of magnitude. And it looks like some of this movement really does need to be accounted for to understand the incidence rates, with significant implications, again, for how you would do monitoring and control in the region. So ran a little longer than I wanted to. Sorry, that's what happens when you let professors talk. But just a few of the next steps here, I break them into 4 categories. We're really working the application here. As I noted, there's been a lot of political turnover in Peru, for those who know the region, which has hampered our ability to operationalize a forecast. So now we're starting to train and transfer to some universities and research institutions in the region rather than straight to the government. We think it'll give us better stability. We're having our first meeting this week on an effort to expand to include Colombia and Brazil. So it's a big upscaling of the effort. And we're also seeing, can we transfer this to an area in Central America, working with the Clinton Health Access Initiative, sorry, split the letters, on Central America, where the case counts are low and therefore the ecology and environmental sensitivity of the system shifts. It seems to cross a threshold. So we want to see how the approach works there. And last, but certainly not least, through these combined methods, but again, all trying to leverage the power of the different fields to understand malaria sensitivities, how can we continue to explain these coupled natural human mechanisms, which, despite the fact that we've known about these relationships since ancient times, we continue to struggle to understand in a predictive manner today. So thank you again for the opportunity to talk. I realize I didn't leave too much time for questions, but maybe we have time for a couple.
Thank you, Ben, for the great talk. So we actually have a class right after seminar, so I think we only have time for one question. Other students have already read the paper that you mentioned, published in IPH. So anyway, you want to ask a question directly? Okay, so let me ask you this question. So Ben, you give a wonderful talk from the importance of value climate variability, the importance of having the data, and then from the very state-of-art sub-seasonal to seasonal forecast. The students, when they read the paper, they have one question regarding actually also COVID-19 related. So do you see how to apply this malarial forecast system, the application to COVID-19 control forecast system?
Yeah, interesting point. So I'm going to answer in a very general way. There are obviously very different diseases, right? We're talking about a vector-based tropical disease versus a pandemic virus with a lot of airborne transmission. But I would say that the general challenge of bringing these different data sets together is really critical, and we can do cross-learning across diseases. Because one thing we've really struggled with in COVID is to bring all the information together in systematic databases for responsible analysis. And we were able to leverage some of the things we've done with malaria and other tropical diseases to build COVID information databases to support research. And I know that Kai did his own work to pull his own database together. So moving forward, how can we use all these diseases to inform those kinds of data structures, I think would be, and cross-learning on approaches would be the way to go. I wouldn't necessarily endorse any single thing that I did here on malaria as the answer for COVID-19 model. They're too different. But if you can really focus on that kind of informed integration, I think there's a lot to be learned.
Thank you so much, Ben. And thank you all guys for coming today. And thank you for our online audience. And just kind of reminder that today's lecture is recorded and will be available online on our center's website, so you can check that. Want to sincerely thank you, Ben, for giving this talk.
Great, thank you.